Redford, Chair of the IUCN Task Force on Synthetic Biology and Biodiversity Conservation. And this presentation is entitled Genetic Frontiers in Conservation, an Assessment of Synthetic Biology and Biodiversity Conservation. When we think about cows, we think about products from cows, leather or meat or milk. But what if we could get cow products without the cows? In fact, products from cow genes have been inserted into algae and then used to produce meat, milk, and leather, all using the tools of synthetic biology. What is synthetic biology? There is not a universally agreed upon definition, but the Convention on Biological Diversity defines it as a further development and new dimension of modern biotechnologies that combine science, technology, and engineering to facilitate and accelerate the understanding, design, redesign, manufacture, and modification of genetic materials, living organisms, and biological systems. Synthetic biology is a field that is being pursued quite aggressively throughout the world, and it is best developed in the areas of agriculture and of human medicine. In agriculture, synthetic biology tools are being used to transform rice from a less efficient to a more efficient form of photosynthesis, to increase the micronutrients in existing crops, create new crops, increase drought tolerance of crops and disease resistance, add nitrogen fixing capacity, a very important fertilizer saver, and as well as a variety of different approaches and applications within the production of domestic animals. The second field in which it's best developed is human medicine, where applications are being developed or deployed to reduce malaria burden, develop new or improved drugs, improve drug effectiveness, improve disease monitoring, as well as improved nutrition. Synthetic biology and its tools have not been heretofore applied to conservation, although there have been a variety of areas in which there has been serious consideration of such application. These include eradicating invasive rodents from islands, controlling invasive mosquitoes to prevent bird extinctions, addressing conservation threats to North America's black-footed ferrets, modifying American chestnuts for potential forest restoration in North America, modifying coral so that they can adapt to climate change and ocean acidification, replacing the blood from horseshoe crabs, which is currently used for biomedical tests, as a way of approaching malaria vector suppression in Africa, and finally addressing honeybee colony collapse. Now, in the World Conservation Congress in Hawaii 4 years ago, the membership passed a resolution that asked IUCN to undertake an analysis of the potential applications of synthetic biology to biodiversity conservation, including the potential application of gene drives. And in the course of this assessment, we considered gene drive one of the synthetic biology tools. IUCN created a task force, of which I'm a chair, consisting of commission members representing natural and social sciences, governments, NGOs, academia, and business, represented from indigenous peoples, as well as many of the world's continents. The task force then appointed a technical subgroup who were actually the lead authors on the assessment, and that assessment is the basis for this presentation. And this technical subgroup included the same kind of broad representation as was present on the task force itself. That group then wrote and published this technical assessment, again, on which this presentation is based, and this is where more details of all of what I've said can be found. And it's available on IUCN's website in English, Spanish, and French. Now, there was a first draft of this that was published and distributed to 13,000 people. We received over 500 comments and then modified the final version based on those comments. And one of the comments that came back from many members was the need to stress the fact that there were both negative and positive anticipated effects of synthetic biology on biodiversity conservation. Now, when you look at the ways that synthetic biology might affect conservation, there are two major streams by which this could take place. On the left-hand side is through applications intended for conservation benefits directly, such as we've discussed heretofore. And then the second big and important stream is through applications not intended for conservation benefits, but that might impact conservation, most particularly, for example, through agriculture. So these two streams meet then in terms of their implications on biodiversity at gene, species, and ecosystems in potential both positive and negative ways. The assessment lays out 10 key messages, and I'm just going to briefly describe them here. First, that there are important conservation implications for these tools that really need to be considered. Second, that conservation needs new tools. The current ones that are being deployed are not sufficient to meet the international goals, both in scope and in pace. Third, that synthetic biology itself as a field is growing extremely rapidly with a great deal of investment throughout the world. Fourth, that engineered gene drive, one of the tools of synthetic biology, is unproven. It's still being tested, but it has great potential, both positive and negative, for conservation. Fifth, that there may be possible but untested beneficial conservation impacts. But sixth, there may also be possible but untested detrimental conservation impacts. Seventh, that the ways in which values and worldviews influence development and assessment of these tools is a critical component to consider. Eight, that indigenous and local communities must be key actors in making research and implementation decisions. Nine, that existing governance structures may be challenged by the applications, but on the other hand, they may be robust enough. And 10th, that risk assessment is not what this assessment does. That needs to be done on a case-by-case basis. For all of these key messages in the assessment, you will find that we classified them in terms of the level of agreement and consensus, as well as the quantity and quality of evidence, giving the reader a better handle for the robustness of the key message. Now, this document, this assessment that we've been discussing and that I've been summarizing, was the basis for the drafting by the task force of a set of principles which were then submitted to the IUCN Council and which developed from that a motion to be put in front of the membership. These draft principles, because they still are draft, are based on a set of principles. That biodiversity conservation is an imperative, that we must consider intergenerational equity as well as sustainable development, and have respect for the rights, beliefs, and cultures of peoples throughout the world, that stakeholder and rights holder participation is critical, as is free, prior, and informed consent, that all decisions should be evidence-based and arrived at through a multidisciplinary dialogue between conservationists and synthetic biologists. And then eighth, of course, that many of the decisions have a very important ethical component that must be raised and considered. Further, that these draft principles, there are a set of key considerations if they were to be applied. First, and critically, is that the decisions need to be made on a case-by-case basis, not a broad scale. Second, existing alternatives to synthetic biology tools must be assessed first and be found wanting before there should be further consideration of synthetic biology applications. Next, that there must be examination for synthetic biology applications that may affect conservation but are not designed for conservation purposes. And agriculture, again, is a particular case for this. That there should be a staged assessment of risks and benefits. That governance must be addressed before all of these are implemented. That there are critical knowledge gaps and research needs, as well as knowledge transfer and capacity building. And lastly, that we acknowledge that there has been some movement for potential introduction of moratoria regarding these applications. Now, where are we in this process? We were to have held the IUCN World Conservation Congress in June 2020 in Marseille, France. I am making this recording in November of 2020, and we are still without a date for the holding or the new session, new convening of the World Conservation Congress. So this policy needs to be voted on by the IUCN membership, and the fact that there has been no Conservation Congress means that there has been no vote. And so we maintain this holding position, and this presentation is a way of reaching out to more people who are interested in the topic and educating them in advance of the anticipated vote. And finally, I finish with a personal observation. That is that given the pandemic and its very large impacts on all aspects of human society and the natural world. That if there is to be a coronavirus vaccine developed through the application of synthetic biology tools, that may change the public's view about the safety or lack of safety of the application of these tools. And this is something only time will tell, but it's certainly worth considering. So thank you very much for your interest in this, and we look forward to your careful consideration and informed debate. Thank you.